Histology Part 1. The histology is total of 17 pages. We will do it in one video. Just listen to it like 2 to 3 times to remember. Epithelium types. There is simple epithelium and compound epithelium. Simple epithelium is one layer. Compound epithelium is multiple layer. Simple epithelium features and they are called simple squamous, simple cuboidal, simple columnar. You can easily remember their names define them. Squamous, length is less than breadth, length equal to breadth in cubes, and length is more than breadth is columnar. Nucleus is flat in squamous, round in cuboidal, elongated in columnar. Function: squamous is for exchange, cuboidal is for secretion, and columnar is also for secretion. Seen in. Now exchange function. So where will you see squamous epithelium? You will see it in Bowman's capsule. If you remember, we draw squamous epithelium in Bowman's capsule. You will see it in blood vessel. We also draw squamous epithelium in blood vessels. Body cavities, you can remember that. Exchanges, body cavities, squamous. Alveoli, we draw squamous epithelium in alveoli. So because we have been drawing the diagrams, it's easy to remember this sites where squamous epithelium are present, like Bowman's, blood vessel, body cavity, and alveoli. Then cuboidal. So cuboidal secretory function: thyroid follicles, germinal epithelium of ovary, and tubules of kidney. The PCTs also has cuboidal epithelium. So thyroid follicle, germinal epithelium, and tubules. Now, columnar, very easy to remember, is the large intestine, uterus, and stomach. Also easy to remember. Stomach and large intestine and uterus has columnar. You can see the diagram. This is a simple cuboidal epithelium, round nucleus, and this is simple columnar epithelium because of the elongated nucleus. Pseudostratified epithelium. Single layer of cells with nuclei arranged at different levels. So they appear multi-layered, but they're actually only a single layer. Appear stratified. So pseudostratified. Pseudostratified ciliated columnar. Seen in trachea and bronchus. Easy to remember because they are a continuous structure. So this special type of columnar epithelium, which is pseudostratified plus ciliated, is present in the trachea and bronchus so that they can sweep up all the discharges and stuff. Compound stratified epithelium. Features. So now compound, that is multiple layer, or which is also called stratified epithelium. What kind of epitheliums are there? So there will be stratified squamous, there will be stratified cuboidal, there will be stratified columnar, and there will be transitional, so also known as urothelial cells. So apical layer, the first layer in stratified squamous, that's the top layer, is squamous. Cuboidal is cuboidal and columnar is columnar. So they are naming it basically on the top layer which is present. And this is a special transitional urothelial cells which has umbrella or inverted U-shaped cells. Transitional urothelial cells. Intermediate layer, the in-between layers are polygonal cells, and basal layers are all columnar. So very easy to remember. It is named on the apical layer. The basal layer is always columnar, and the in-between layers are polygonal. So where are they seen? Stratified squamous, easy to remember, is skin because we have been drawing it many times. It is also present in tongue, tonsils, esophagus, lower part of anal canal, vagina. This is asked in MCQs. Lower part of anal canal is stratified squamous. And vagina also is stratified squamous. So I think if you listen to the video 2-3 times, you will be able to remember that stratified squamous epithelium is present in skin, tongue, tonsils, esophagus, lower part of anal canal, important, and vagina. Stratified cuboidal presence in ducts of exocrine glands and sweat glands. Stratified cuboid epithelium present in exocrine glands and sweat glands. And stratified columnar is present in membranous and penile urethra. There are different parts of urethra, so membranous and penile urethra has stratified columnar. And transitional urothelial cells is present in ureter, urinary bladder, prosthetic urethra. So regarding the urine system, has transitional or urothelial cells where the top cells are U-shaped. Ureter, urinary bladder, prosthetic urethra. Apical layer squamous, flat nucleus, intermediate layer polygonal, different shapes. Basal layers is columnar. This is a stratified squamous epithelium. Transitional epithelium, urothelial cells. Apical layer are umbrella or U-shaped, if you can see. Intermediate layer are polygonal and basal layers are columnar cells. Surface projections. Microvilli increases surface area, seen in small intestine for absorption and gallbladder for storage. Crypt and villi, microvilli. So microvilli is this hair-like projections on the cells. Enterocyte. Lumen of small intestine has microvilli. Microvilli is present in small intestine and gallbladder for storage, and we learned something about large intestine. Which was that it is lined by simple columnar cell. Cilia, function, motility. Simple ciliated columnar seen in eustachian tube, middle ear, ventricles of brain, fallopian tube. Cilia's function is motility. So here cilia is actually moving, but villi is, I think, static. So simple ciliated columnar seen in eustachian tube, middle ear, ventricles of brain, and fallopian tube. Pseudostratified ciliated columnar seen in trachea and bronchus. Glands. Secretory epithelium invaginating into connective tissue is called glands. Secretory epithelium invaginating into connective tissue is called glands. Endocrine versus exocrine glands. Endocrine glands and exocrine glands. Exocrine glands has this ducts and secretory part. Secretes hormones, endocrine system. So endocrine gland secretes hormones. Exocrine secretes enzymes. Ducts are absent in endocrine, present in exocrine. Secreted via bloodstream. Secreted via ducts. Vascularity, endocrine more than exocrine. Because Endocrine secretes via blood. Goblet cells. Unicellular glands are called goblet cells. Secretes mucus via exocytosis. Mucus is secreted through opening into airway. Goblet cells appears pale on H&E stain. Types based on number of ducts. Simple glands secretes into single duct. Compound glands secretes into multiple ducts. So based on number of ducts, it can be simple or compound. Single duct is simple and multiple ducts is compound. Diagram. Simple ducts. Simple alveolar acinar not found in adult. Gland cells, duct surface epithelium. A stage in development of simple branched glands. Simple branched alveolar, which are sebaceous or oil glands. Simple tubular intestinal glands. Simple coiled tubular merocrine sweat glands. Simple branched tubular gastric glands and mucous glands of esophagus, tongue and duodenum. Compound alveolar acinar mammary glands. Compound tubulo-alveolar. Salivary glands, glands of respiratory passage and pancreas. Compound tubular. Mucous glands in mouth. Bulbourethral glands in male reproductive system and testis. Seminiferous tubules and mucous glands in mouth. Based on type of secretions, microscopy. Lumen small, nucleus round, pyramidal cells. Serous glands. Mucous glands. Lumen large, nucleus flat, pushed towards base, columnar cuboidal cells. Secretions watery, secretion thick. Apical eosinophilia present due to secretions collected towards apex, absent on mucous glands. Example, parotid glands, sublingual glands. This is easier to remember. If it is sublingual, obviously they will release mucus. Parotid, you can remember that it has a thin secretion, so parotid glands are serous. Note, submandibular gland is mixed gland. Serous plus mucinous acini. Based on mode of secretion, you have merocrine, apocrine and holocrine. Mechanism of secretion. Secretion, secretory vesicule, nucleus, Golgi complex. Exocytosis. Pinched off portion of cells is the secretion. Apex of cell detaches. Pinched off portion is equal to secretion. Mature cell dies and becomes secretory product. Whole cell disintegrates, has multiple layers. Holocrine glands. Mature cell dies and becomes the secretory product. Whole cell disintegrates, has multiple layers. Basal layer is mitotic. Regenerates disintegrated cells. Cell is intact in merocrine glands, not intact in apocrine, and not intact in holocrine. So only intact in merocrine, who secretes the products into the duct. Example of merocrine gland is exocrine glands and sweat glands. Apocrine example is mammary glands. Atypical sweat glands of axilla and pubis. Holocrine glands are sebaceous glands. Connective tissue provides support to organs, provides passage for neurovascular structures, connects adjacent layers. Types: general connective tissue and special connective tissue. Components: cells and matrix. Cells are adipocytes, fibroblasts, and macrophages. Matrix: fibers and ground substance. Reticular fibers, adipocytes, mesenchymal cells, elastic fibers, collagen fibers, fibroblast, macrophage, fibrocyte, adipocyte, mesenchymal cell. Connective tissue. Fibers. Fibers. Collagen fibers. Long wavy fibers present in bundle, appears white to naked eye, that are collagen fibers. Appearance on staining: H&E stain, which is hematoxylin and eosin stain, and Van Gison's stain appears pink, the collagen fibers. Mason trichrome stain and silver impregnation stain. So Mason trichrome appears blue, silver impregnation appears brown. Type: collagen type 1, 2, 3, 4. Type 1 found in ligaments, fascia, tendons, aponeurosis, dermis, fibrocartilage. 2, only one place, hyaline or elastic cartilage. And 3, reticular fibers, lymphoid organs except thymus. 4, lens capsule and basement membrane. So I think it's easier to remember 2, 3, 4 because they're only present at 2, 1 or 2 places. So type 2 is present at only one place. Type 2 collagen fiber, which is hyaline or elastic cartilage. Type 3 is reticular and it's present in all lymphoid organs except thymus. And type 4 is present in the lens and basement membrane. Reticular fibers, short fibers, branch anastomose and form reticulum network. Appearance on staining black. Reticular fibers on silver impregnation, black brown. Reticular fiber, short fibers, branch anastomose and forms a reticulum network. Elastic fibers, short non-wavy single fibers not present in bundles, thin except in ligamentum flavum. Appearance on staining, H&E stain. Here are the elastic fibers. Orcine with aldehyde fusion. Cells. Fibroblasts, adipocytes, intrinsic component, macrophages, immune system cell. Cells, fibroblasts, adipocytes, intrinsic component and macrophages are the immune system cells. Spindle shaped cells, flat nucleus. Fibroblast, what are that? Fibroblast, most common cell of connective tissue. Most common cell of connective tissue. Fibroblast. Functions, synthesizes extracellular matrix and collagen, forms skeletal framework for tissues, helps in wound healing. Very important cell. So until now I thought fibroblast must be doing nothing, but it actually makes the extracellular matrix and collagen fibers, and forms skeletal framework for tissues and helps in wound healing. And collagen, we here saw that collagen fibers are again of different types. Adipose tissue, aggregation of fat cells is called adipose tissue. Types, white or yellow adipose tissue and brown adipose tissue. White or yellow is unilocular cells, nucleus pushed to periphery. Brown adipose tissue is found in newborns, is multilocular cells. Staining on H&E appears as empty space. Xylene and benzene dissolves fat. Special stain is Sudan stain for the fat. Sudan stain. Note, based on arrangement of fibers, there is dense regular fibers and dense irregular fibers. Dense regular fiber is present in aponeurosis, ligaments and tendons. Collagen fibers, fibroblast nuclei. Aponeurosis, ligaments and tendons, all the tight and strong structures has dense regular fibers. Dense irregular fibers is seen in dermis of skin and organ capsules. Dense irregular fibers are seen in dermis of skin and organ capsules. Organ capsules and dermis of skin has dense irregular fibers. So these are the fibroblast nuclei and the collagen fibers. Moving to part 2, cartilage. This is a special connective tissue. Cells in it are called chondrocytes or chondroblasts. Matrix, ground substance plus fibers. Types, hyaline cartilage, elastic cartilage and fibrocartilage. So, a special connective tissue is your cartilage. The cells are called chondrocytes or chondroblast. Matrix has ground substance plus fibers. So, chondrocytes arrangement. In hyaline and elastic cartilage, it is in groups, while in fibrocartilage, it is in rows. So, here there is a diagram, matrix. Appearance of hyaline is shiny, glossy. Refractive index of type 2 collagen fibers similar to that of ground substance. That is the reason why it is shiny. Here is the cell nest, lacuna, chondrocyte, chondroblast, outer fibrous layer, inner cellular layer, territorial matrix and interterritorial matrix. Elastic cartilage, elastic fibers. Type 2 collagen. So we read that type 2 collagen is present in hyaline and elastic cartilage. And all the other things are in type 1. So there you can see chondroblast, chondrocyte, lacuna, elastic fibers, inner cellular layer, outer fibrous layer. Coming to fibrocartilage, type 1 collagen, long wavy bundles, chondrocyte, collagen fibers, lacuna. And both in hyaline and elastic cartilage, you have outer fibrous layer plus inner cellular layer, which forms the perichondrium. So both in hyaline and elastic cartilage, you have the outer fibrous layer and inner cellular layer called perichondrium. So perichondrium present in elastic cartilage, hyaline cartilage, but absent in fibrocartilage. Examples: hyaline cartilage. Hyaline cartilage is present in articular cartilages, costal cartilages, embryonic cartilages, epiphyseal cartilages, and respiratory cartilages. So you'll have to hear it 2-3 times to remember that hyaline cartilage, the shiny glossy cartilages, are present in articular, which we have seen with our naked eyes, costal, embryonic, epiphyseal, and respiratory. Whereas elastic cartilage is present in epiglottis, earlobe, eustachian tube, corniculate cartilage, cuneiform cartilage, apex of arytenoid. And fibrous comes in meniscus, articular disc, glenoidal labrum, and intervertebral disc. Only hyaline cartilage can ossify. Important. Only hyaline cartilage can ossify. So which are those? Articular, costal, embryonic, epiphyseal, respiratory. Muscle made up of myocytes, elongated muscle fibers, covered by connective tissue, provides pathway for neurovascular structure. Covered by connective tissue, provides pathway for neurovascular structure. Skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is in skeletal system. Striation is present. Voluntary muscle supplied by somatic nerves. Cardiac muscle present in heart, striation is present. Involuntary muscle, autonomic nerve supply. Smooth muscle is present in hollow viscera. Striations are absent in voluntary autonomic nerve supply. Covering in skeletal muscles. Muscle fiber. A group of fibers, fascicle, covered by endomysium, covered by perimysium. The connective tissue like thing is endomysium, which is collectively packed in perimysium. In between you'll find blood vessels and epimysium, and then bone, tendon. Coverings in a skeletal muscle. Skeletal muscle is collection of fasciculi. Coverings, epimysium. Outermost layer of a muscle is epimysium, encloses numerous fasciculi. Perimysium, covering of each fasciculus, encloses numerous muscle fibers, that is perimysium. Endomysium, covering of each muscle fiber. So the most basic unit is endomysium, which is covering each muscle fiber. Endomysium. Bundle, perimysium. Entire muscle, epimysium. Endo is the smallest, epi is the biggest. Skeletal muscle layer. Long cylindrical fibers arranged parallel to each other. Varies in length and diameter. Each fiber is a syncytium with multiple hundreds of nuclei. Nuclei are flat, elongated, peripheral. Skeletal muscle H&E stained. Features: long cylindrical fibers arranged parallel to each other. Varies in length and diameter. Each fiber is a syncytium, which means multiple nuclei. Nuclei flat, elongated, and peripheral. Sarcomere. A band is the dark band, I band is the light band. Z line is the dark line in I band. A band is the dark band, I band is the light band, Z line is the dark band in I band. Sarcomere. Cardiac muscle. Arrangement of fibers is not parallel. Fiber gives branches which anastomose with each other. Nucleus central and spherical. No syncytium. Intercalated disc present. So there is no syncytium, one single nucleus, central, spherical, intercalated disc present. Smooth muscle. Striations, no striations. Spindle shaped cell. While this, they have branches, the cardiac muscle. Nucleus is oval. It's circular, central, spherical in cardiac muscle. Lymphatic system, lymphoid organs. Primary and secondary. Primary and secondary lymphoid organs. In the lymphatic system, primary. Produce lymphocytes independent of antigenic stimulus. For example, thymus and bone marrow. So lymphoid organs are primary and secondary. So primary are doing their job without any stimulus, like they keep producing cells. So that is production of lymphocytes without any antigenic stimulus. That is primary lymphoid organs, which are thymus and bone marrow. Secondary produces lymphocytes in response to antigenic stimulus. So those are spleen, lymph nodes, tonsils, mouth, gout. So they need antigenic stimulus to produce lymphocytes. So they can produce lymphocytes, and these are spleen, lymph node, tonsils, mouth, galled. Lymph node parts. Outer convex part has openings of numerous afferent lymphatic vessels. Outer convex part of a lymph node has openings of numerous afferent lymphatic vessels. Subcapsular sinus present below capsule. Identification feature is subcapsular sinus. What is the identification feature of a lymph node? Subcapsularis sinus. Outer cortex. Lymphocytes arranged in follicles. Dark periphery with numerous lymphocytes. Inner pale germinal center. Dark periphery with numerous lymphocytes. Inner pale germinal center. Inner medulla. Lymphocytes arranged as cords. Inner medulla. In lymphocytes arranged as cords. Flow of lymph. Efferent vessels. Subcapsularis sinus. Trabecular sinus. Medullary sinus. Efferent vessels. Here is the diagram. Dark periphery, inner pale germinal center. Lymph node follicles. Dark periphery, inner pale germinal centers. Efferent lymphatic vessels goes to subcapsularis sinus, trabecular sinus, medullary sinus, and efferent lymphatic vessel. Spleen parts: white pulp and red pulp. White pulp is lymphocytes arranged as follicles. Eccentric arterioles. Red, that's the white pulp, and red pulp is splenic cords of Billroth. Contains reticular fibers, fibroblast, and macrophages. The splenic cords of Billroth and sinusoids, lined by stave cells, banana-shaped endothelium. Spleen parts: white pulp, red pulp. White pulp has lymphocytes arranged as follicles and eccentric arterioles, and red pulp has splenic cords of Billroth, which has reticular fibers, fibroblasts, macrophages, and sinusoids, which is lined by stave cells, banana-shaped endothelium. Spleen, capsule, central artery, trabeculae, lymphatic nodule. White pulp, trabecular vein and artery, lymphatic nodule. White pulp. Central artery, germinal center, central artery, trabeculae, splenic cords in red pulp, venous sinuses in red pulp, pulp arteries. Tonsils features: tonsillar crisps. Identification features: tonsil feature, tonsillar crisps, which is the identification feature, like here, tonsillar crisps. Stratified squamous non-keratinized epithelium. Lines oral surface. Only lymphoid organ with epithelial lining. Lymphocytes arranged in form of follicles. Tonsillar crypts is the identification feature. Stratified squamous non-keratinized epithelium. Lines the oral surface. Only lymphoid organ with epithelial lining. Lymphocytes arranged in form of follicles. Lymphocytes were arranged in form of follicles also in spleen. Thymus, primary lymphoid organ. Thymus and bone marrow. Features, they have a capsule. Lymphocyte arranged in lobules, not follicles. Lobule, outer cortex, numerous lymphocytes, inner medulla, fewer lymphocytes with Hassel's corpuscle. Lobules. So in thymus you will find lobules, outer cortex and inner medulla. Outer cortex, more lymphocytes, numerous. Inner medulla, fewer lymphocytes with Hassel's corpuscles. Few lymphocytes with Hassel's corpuscle. Concentrically arranged eosinophilic mass. Concentrically arranged eosinophilic mass. Degenerated cells. Concentrically arranged eosinophilic mass. Arranged eosinophilic degenerated cells. Last part, histology part 3. Histology of the GIT, layers, mucosa, lamina propria, muscularis interna, and submucosa, muscularis externa, serosal or adventitial layer. Starting with mucosa, epithelium. Epithelium up to esophagus, stratified squamous, non-keratinized. So in the GIT up to esophagus, the lining is stratified, squamous, non-keratinized. Coming to stomach, so up to esophagus, even the esophagus has this stratified, squamous, non-keratinized. Stomach changes to simple columnar. Small intestine, simple columnar with microvilli. Large intestine, again simple columnar. In the large intestine it is simple columnar, but in the anal canal below the pectinate line it changes to stratified squamous, non-keratinized. Hilton's white line, and in anal verge it goes to stratified squamous, keratinized epithelium. So GIT up till esophagus, stratified squamous, non-keratinized. Stomach, simple columnar. Small intestine, simple columnar with microvilli. Large intestine, simple columnar up till pectinate line, then stratified squamous, stratified columnar, non-keratinized. After Hilton's lines on the anal verge, stratified squamous, keratinized epithelium. Lamina propria, connective tissue. Connects epithelium to muscularis interna. Lamina propria connects epithelium to muscularis interna. Content: pairs, patches, lymphatic aggregation in ileum. Content. Lamina propria connective tissue connects epithelium to muscularis interna. Content pairs patch. Lymphatic aggregation in ileum. In ileum, it has lymphatic aggregation called Peyer's patches. Also contains gastric glands and intestinal glands. Muscularis interna. Inner circular muscles, outer longitudinal muscles. Inner circular muscles, outer longitudinal muscle. Submucosa. Strongest layer of GIT. Submucosa. Submucosa is the strongest layer of GIT. Composition, connective tissue. Esophageal glands. Brunner's glands in duodenum. Meissner's plexus regulates secretions and blood flow. Submucosa is the strongest layer of GIT, contains connective tissues. And in esophagus, it has esophageal glands. In duodenum, it has Brunner's glands. And it has Meissner complexes throughout, which regulates secretion and blood flow. And here you can see there in ileum, there is Peyer's patches in lamina propria. Muscularis externa. Muscles made up of smooth muscles, inner circular, outer longitudinal. In esophagus, upper 1/3, middle 1/3, and lower 1/3. In esophagus, upper 1/3 is skeletal, middle 1/3 is skeletal plus smooth, and lower 1/3 is purely smooth. Plexus mesenteric Auerbach's regulates motility. Plexus mesenteric and Auerbach's regulates motility. Serosal or adventitial layer. Serosal covered by peritoneum. Adventitial not covered by peritoneum. We have almost reached the end of histology. So serosal, which is also known as adventitial layer. Serosal covered by peritoneum. Adventitial not covered by peritoneum. Histology of stomach. Pits, glands, surface epithelium, lamina propria, muscularis mucosae, submucosa, strongest layer, muscularis externa, oblique layer, circular layer, longitudinal layer, serosa, gastric pit, gastric glands. Here is the gastric gland pit, isthmus, neck, body, fundus. Stem cells, neck cells secrete mucus. Oxyntic parietal cells, acidophilic cells with round nucleus, secretes HCl and intrinsic factor. Argentafin cells, neuroendocrine cells, have high affinity for silver salts, secrete gastrin. Peptic chief cells secrete pepsinogen. Oxyntic cells, parietal cells, acidophilic cells with round nucleus secretes HCl and intrinsic factors. Chief cells, peptic cells, secretes pepsinogen. Stomach has cardiac end, fundus, pyloric end, body. Cardiac end cells, neck cells, mostly shallow gastric pit, has cardiac glands. Fundus, fundic gland, cells all type, stem, neck, chief, and oxyntic. Here we saw what kind of cells they are talking about. Stem cells are at the isthmus, neck cells secrete mucus, oxyntic cells, parietal cells secrete HCl and intrinsic factor. Argentefin cells, neuroendocrine cells secrete gastrin, and peptic chief cells secrete pepsinogen. Pyloric and pyloric gland cells, neck cells, which secretes mucus, and argentefin cells, which secretes gastrin in the pyloric end, deep gastric pit, body, fundic glands. Fundic glands has neuroendocrine argentefin cells, gastrin secretion, peptic and chief cells, pepsinogen secretion. Note, cell and fundus of glands, chief cells and argentefin cells. Argentefin secretes gastrin, chief cell secretes pepsinogen. Cells and fundic glands. That's in the fundus of the glands, which is here. Cells in fundic glands, all type of cells. Goblet cells are absent in stomach. So neck cells secretes mucus cells, but goblet cells are absent in stomach. Histology of liver, hepatic globule. Central vein, portal triad. Space of mole, periportal space, portal vein, bile duct, hepatic artery, space of Deci, sinusoid, hepatocytes, anastomosing cords, one cell, 2 cell thick, 1 or 2 cell thick, hepatocytes, anastomosing cords. This is a hepatic lobule. Central vein, portal triad. Blood flow, portal vein, hepatic artery, opens and sinusoid, central vein, hepatic vein, IVC. Hepatic lobule cross section, central vein, portal triad, portal field, hepatic arteriole, hepatocytes, hepatic lobule, endothelial cells, Kupffer cells, sinusoid, space of Deci. Endothelium, hepatocytes, Kupffer cells in sinusoids, space of Deci between hepatocytes and sinusoids contains stellate cells, Ito cells. Gaps between endothelium for transport of substance between hepatocytes and sinusoids. Portal lobule, hepatic lobule, central vein, corner. Portal lobule, triangular. Signifies direction of bile flow, centropital. Portal triad, center. Portal lobule. Hepatic acinus. Zone 1, 2 and 3. Highly vascular, zone 1, most commonly affected by toxins. Zone 2, less vascular. Zone 3, least vascular, prone to ischemic injury. Hepatic acini. Diamond shaped, area between 2 adjacent hepatocytes. Supplied by portal vein and hepatic artery. Histology of respiratory tract. Trachea and bronchus is turo-stratified ciliated columnar epithelium. trachea, bronchus, primary bronchus, secondary bronchus, tertiary bronchus, pseudo-stratified ciliated columnar epithelial. Bronchioles, simple columnar, simple sedated columnar epithelium. Terminal bronchioles, respiratory bronchioles, simple cuboidal epithelium. Alveolar duct, alveolar sac, alveoli, simple squamous epithelium. Histology of trachea and bronchus. Pseudo-stratified ciliated columnar epithelium, cilia, goblet cells. Submucosa contains submucosal cells. Cartilage layer, paired chondrocytes, matrix hyaline. Bronchioles, simple columnar, simple ciliated columnar epithelium, cartilage replete by smooth muscle. Note, goblet cells, submucosal cells are absent in bronchioles. Goblet cells are also absent in stomach. Smooth muscle, bronchioles, simple columnar connective tissue. Alveoli cells. Cell type, type 1 pneumocyte 95%, type 2 pneumocyte 5%. Cell type, type 1 simple squamous, type 2 pneumocyte large cuboidal cells. Function, gas exchange, secrete surfactant. Alveoli sac. Type 1 pneumocyte 95%, type 2 5%, simple squamous, large cuboidal cells. Secrete surfactant. That's the end of histology.